Our next presenter, we're happy to have here Dr. Lawrence Korngut, who's a neurologist, the head of the Calgary ALS and Motor Neuron Disease Clinic, and associate professor of neurology at the University of Calgary, and the principal investigator for the Canadian Neuromuscular Diseases Network, as well as the Canadian Neuromuscular Disease Registry. He's also a member of ALS Canada's Scientific and Medical Advisory Council, and is the recipient, the lead recipient of a team that received a 2015 Arthur J. Hudson Translational Team Grant. Please welcome Dr. Lawrence Korngut.
Well, thank you very much for the kind introduction. It's wonderful to be with you all here today. It's great to see such a great turnout, and we're right on time. So let's begin. I'm going to be talking about this clinical trial of Pimozide for ALS that we've been working on, and one that we've already completed. And I think this is very exciting work because it's coming out of a truly Canadian story of discovery in terms of possible treatments for ALS. And of course, it's hard to summarize all of this in 15 minutes, but I'll certainly do my best. I don't have any conflicts or disclosures for this work. We always have to declare as researchers whether we have pharmaceutical industry involvement in treatment development, and here there really is none. And one of the exciting things about Pimozide relates to that, in the sense that this is a treatment that's already been available for over 50 years in Canada, and it's quite inexpensive. So just to illustrate what we're dealing with here in terms of the cost of a drug like this, because it is a generic, when you look at the new treatments out there for other diseases that are new and exciting, nusinersen is a new treatment for spinal muscular atrophy, and it's expected to cost about $750,000 US a year. Adalurin is the new treatment for Duchenne muscular dystrophy at a cost of about $500,000 a year. And Pimozide, of course, is about $100 a year. And so there's challenges in terms of developing these kind of treatments that there is no pharmaceutical company interest in, because there's really no profit to be made off of a drug like this. But it also emphasizes why it's so important and why we're so grateful to the support of ALS Canada and Brain Canada for this research, because it really allows us to look at the best opportunities out there to find treatments, even when the medications are of very low value. And so I think it's important to highlight that. As mentioned, this is really a Canadian success story. Alex Parker at the University of Montreal and Pierre Drapeau, along with colleagues, first identified Pimozide as a potential therapy for ALS, and as you'll see, there are other colleagues involved along the way. And basically, the way that they developed this was they inserted some of the genes for ALS into zebrafish, as well as worms called C. elegans worms. And then what they were able to do was place them into these little dishes. So each of the little wells that you see there it has one fish inside, and then these fish, of course, stop moving over time because they develop ALS. What they then did was put through a number of different drugs, really, over 3,800 different drugs into these wells to see whether any of them could reverse ALS, whether they could make these fish move for longer or preserve their mobility, and the same went with the worms. And what was interesting was they found 21 that accomplished this, and 12 of these 21 had very similar mechanisms of action, and these were all neuroleptic medications which are used for the treatment of psychosis, so diseases like schizophrenia, diseases where individuals are hallucinating or they're delusional, and so of course these were used by psychiatrists exclusively for that purpose. But it was quite interesting that these seemed to show up on this panel. At that point, I had met these researchers through the ALS Canada Forum, which was a nice way to meet people. I was seeing some of the research being presented, and we decided to just go ahead and then try Pimozide, which is already available as a generic medication in a clinical trial. And so we began this at the University of Calgary a number of years ago. where we compared the patients who received, and you'll see the different groups there, who received 6 milligrams of Pimozide versus 10 milligrams of Pimozide, versus a group of placebo patients. Now, this was a very small study. This was only 10 patients at the lower dose of 6 milligrams, 10 patients at 10 milligrams, and 5 patients in the placebo arm, and we'll come back to that. I mean, that's a very small placebo group, but we weren't sure what to expect, and mainly what we wanted to learn from this very small study, you know, it's very, I don't think you can ever really prove a treatment works with this small of a sample size of patients, but what we were hoping to learn was how this drug affected individuals with ALS, because of course individuals with ALS are very different from individuals with psychiatric conditions, and so keep in mind that the prime goal of this was to find the right dose. So we had designed it that the doses could be decreased over time if individuals were not tolerating it, and as you'll see, that's exactly what happened. There's a lot of details on these slides, but this slide is just to show that at the top we ended up seeing 38 patients, 25 of them qualified, and most finished. So 5 of the placebo patients finished, all 5, and 19 out of 20 of the Pimozide patients finished. The challenge that we came across, however, was when we do a randomized controlled trial, we randomly put people into groups, and if there's enough people and randomization is successful, all 3 groups look the same before any patient is administered a medication, and that's really a critical component of clinical trials. So the first thing we look at when we look at results of a clinical trial, it's called our table 1, where we look at what did the placebo group look like, the low dose and the high dose. And you'll see here the low dose has already changed from 6 milligrams down to 2 milligrams, and the high dose we reported as the patients who received 4 milligrams, 6 milligrams, and 10 milligrams. And I'll come back to that. But the first thing you notice is that in the patients in the placebo group, they were actually more severely affected. And so just by chance, and again, because there's only 5 of them, when you have such a low number of patients, you can get these imbalances between the groups. But they had their vital capacity. Unfortunately, here it is. Here's my cursor. So as you can see, you know, they were a little bit younger, the patients in the placebo group compared to the treatment groups. Their ALSFRS score was lower, indicating they had less function, so they were more advanced in their disease. Their vital capacity was lower at 68% compared to the other 2 groups, which were closer to 90%. There was a higher percentage of bulbar onset, which we know does progress faster on average than limb onset. A higher number on BiPAP. Oh, I've lost my cursor. On BiPAP, and of course, when we measured strength, their strength was lower. So any conclusions that we come up with in terms of trying to interpret this initial clinical trial have to be tempered by the fact that the placebo group was more advanced. So this placebo group was progressing faster even before the patients started in the trial, meaning that much of the effect that we see in this trial may not be that the drug works, but it may be that the placebo group was just moving faster in terms of progression. So that's important. So this really makes us unable to jump to any conclusions as to whether the treatment will work. But I will show you the data, but we just have to keep that in mind. As we went through the clinical trial, it became very evident that the higher doses of 6 to 10 milligrams were just not tolerated. And I think this is the real key point to take away from this first trial, was that we figured out that really anything above 4 milligrams was not tolerable by individuals with ALS. And so for the next trial, which I'll be speaking about shortly, we're sticking to a 4 milligram dose with the possibility to reduce it if we need to in a given individual, but we're not going to go higher than that. And I think that's an important result of this first trial. So what did we learn? Well, we learned that patients on Pimozide can sometimes feel more weakness, in terms of not that they become weaker from the drug, but they just can feel a bit weaker. And this was in a fairly low proportion of patients, but something to keep an eye on in our next study. And then the major side effect of Pimozide, and this was known previously, is called akathisia. And akathisia is a condition where people just feel restless, feel a little bit irritated and restless, and we know that at 4 milligrams per day, that occurred in about 62.5% of individuals above and beyond the placebo. because of course they're compared to the placebo group, and 25% at the 2 milligram dose. However, most of the time this was very mild, and the individuals on the medication were okay with it. So again, this can range from very mild to very severe. When it becomes bothersome, we do reduce the dose or discontinue the treatment. And as you see, 0% of the 5 patients with the placebo developed this side effect. So it's kind of a unique side effect and usually occurs with these antipsychotic medications. So just to go over the results, we looked at functional rating scale. Keep in mind, these individuals were on the study drug for 6 weeks or the placebo for 6 weeks. So it's a very short period of time. You know, we didn't see any difference between the placebo group, the low dose group, or the high dose group, and we wouldn't really expect to over such a short period of time. In terms of the breathing, you know, there was a bit of a trend of maybe a bit of improvement in breathing at the high dose. However, you know, again, these are very small numbers. The error bars overlap, and so ultimately we didn't see any change there. When we move to the MRC score, which is a measure of strength, we did see quite a significant difference between the low dose group and the high dose group, where there was really no change over time, no worsening of strength, and there was a very significant worsening of strength in the placebo group. And this was statistically significant. But the challenge, however, is that initial issue is that this group was progressing faster, and we know that now. And so could this all be just from the natural progression, that these were progressing faster than these individuals? And absolutely, that can be the case. And so this does not mean that this prevented, the medication prevented progression. But again, it is promising in terms of we do need to look at this further in a larger group of individuals. We also looked at what's called repetitive nerve stimulation, where we zap a nerve. So here you see in the placebo nerve, we zap a nerve, we collect the electrical, measure the electrical activity from the muscle, and you can see this drop off. And with Pimozide, this seemed to be protected. So as individuals' muscles get weaker, this tends to get worse. And we saw this in the placebo group, which of course fits with the strength getting worse. But again, same issue. This group was progressing faster, and that may well account for this difference. And so that's what we found. So we know that over 4 milligrams per day, it's not tolerated by individuals with ALS. We know that there may be some benefit within all of this, but we don't know that exactly, and we certainly can't prove it at this point in time. So what do we do next? Well, now that we know the right dose that we need to use, we're moving on to a randomized controlled trial of Pimozide in 100 subjects across Canada, and as you'll see, there's 9 different university centers, and so this is the clinical trial that we've called Pimozide 2. So again, we're going to be looking at Pimozide on the outcome measures of ALS progression and some electrical outcome measures as well. Instead of 6 weeks on medication, this time it's going to be 26 weeks on the medication, and then we have an observational phase afterwards that through the registry, which is optional for patients to participate in. We can track function upwards of 5 years to see if there's any difference later on based on this treatment. And that will be through the Canadian Neuromuscular Disease Registry. So for those of you who are interested in potentially participating or getting more information about this clinical trial that we expect will be opening for recruitment in the next number of months, certainly by end of October, we expect to be up and running. And of course, it takes time to get all the sites up and recruiting patients, but we expect by end of October, hopefully we'll have everything in place. Right now, the application is at Health Canada and at our ethics committee here at the University of Calgary to get this approved. And so sometimes there's a little bit of change that needs to be done based on that feedback, but we expect that things will move forward rather quickly after that. And so individuals, we do have specific criteria for these things, but essentially to participate, you need to have a diagnosis of ALS and meet the criteria for that. You need to be able to understand and sign the consent form. You have to be an adult over age 18, and onset of muscle weakness had to begin within the last 4 years, so 48 months, which is quite a big interval. Many clinical trials is 24 to 36 months at the most, but we decided to open this up even further out to 48 months. Otherwise, vital capacity has to be greater to than equal to 50%, which of course has to be on the day of the screening visit on the equipment that we're using, and individuals have to be able to swallow tablets or capsules whole at the beginning, and we have to be able to expect that they'll be able to swallow them throughout the study. And then we do have a host of laboratory testing that we do to make sure there are no contraindications. Pimozide does have other side effects, which I didn't mention here today, and it can interact with other medications quite easily and cause quite significant side effects through the other medication or through itself. And so we're very particular about what people can be on while they're on Pimozide, and so individuals have to pass all of these steps, and this is standard for any clinical trial. Also, if somebody's taking Riluzole, they have to be on a stable dose for 30 days. And since Radicava has become available in the United States, and we are hopeful, of course, and you'll be hearing more about this today, sometime within the next year. In Canada, if an individual is receiving Edaravone, also known as Radicava, they are eligible to go on to enter the clinical trial. But they just have to be on 2 cycles of infusion treatments prior to the randomization visit. Once they're on the clinical trial, there also is an option to start Edaravone, so we would not ask people to not go on it. They're able to take that and participate in the clinical trial. We just have to do an extra visit to get some more measurements of things. We also exclude individuals with certain conditions such as akathisia. So if somebody already has this problem, certainly adding a medication like Pimozide is only going to make things worse. So individuals with conditions that can be caused by Pimozide, we're not allowing into the trial. So for example, restless leg syndrome, because that can really be worse, and we saw that in the first trial by this treatment, or any other previous reactions to medications like that. So there's quite a list of these. I'm not going to go through all of them in great detail today. Here's a list of the participating sites that will be joining us for this clinical trial, as long as all goes well. We still have to go through contracting processes and that, but we expect all of these, are the vast majority, will of course be recruiting patients. So if you're interested, these are the sites that you could go to for the trial. And of course, in summary, again, this is a very exciting story, I think, of real strong basic science with the individuals you see listed there. Dr. Zinman and I then took it from there and developed this first trial, and now this second trial. And so it's a nice bench to bedside story. We have a large University of Calgary team that, of course, works on this, and as you can imagine, the majority of work is done by them, and so to them I'm very grateful as well. And of course to our funders. So the first clinical trial of 25 subjects was funded by a very generous donation from the Quirk family, originally here from Calgary, as well as University of Calgary pilot funding dollars, and of course that allowed us to finish that first trial. This next study, we're very grateful to ALS Canada and Brain Canada for the Arthur J. Hudson Translational Team Grant from 2015 to now move forward with this clinical trial. It does take time, of course, to finish the first trial and get everything organized for a big effort like this. So we're very, very excited to get started.
Okay. Great, thank you, Lawrence. I'm sure that could have easily been a 1-hour talk, given if you'd come through how much work went into getting to the Pimozide, and then racing through that in 15 minutes is not easy. I have one question. We have time for about one question. And one of the things you mentioned was that Pimozide's very cheap. It's a generic drug now. We're looking at in ALS potentially a situation where there may be some very expensive potential therapies coming down the pipeline eventually. Do you think that there might be other Pimozides out there? Lots, there was 3,800 compounds in that screen. Could there be, tens of thousands of other screens out there where there might be something that could be beneficial and actually quite cheap?
Absolutely. You know, I think this is a good example of how the process can work and work fairly quickly. You know, I mean, this is a period of about 4.5 years from when this discovery was made through to a fair-sized clinical trial like we're talking about here. But absolutely, I mean, no one treatment may not work for everybody, so it's important to pursue as many as possible. But also, combination treatments, may be the way to go. And so any of these treatments that we can discover and through a process like this, I think is optimal. And Dave, I think you're absolutely right. That this should be repeated, and it's certainly being repeated in other by other teams with focusing on different drugs. But the fact that ALS Canada saw this and felt that this was worth funding, I think says a lot, about ALS Canada's commitment to this kind of process, which I think is absolutely the way to go. And then I would add one more thing. These types of medications, in early clinical trials, can show some excitement. As you saw, if you look at the data one way, you can say, wow, this really works. But actually, when you dig down deeper and you figure out that placebo group was actually progressing faster even before the clinical trial, it really tempers the results and really shows you can't come up with a real conclusion here. You just need to keep working on it. And so I want to be clear, I would not recommend anybody with ALS going on Pimozide at this time. It is a challenging medication to administer. It has a lot of safety monitoring requirements, and I think within the safety of a clinical trial where we're very intensive about these things, it's very reasonable and very safe. But certainly, I don't think there's near enough evidence at this moment to be prescribing it. So just to be clear about that.
Well, thank you. Thank you very much, Lawrence. And we really appreciate it. And we'll talk to you again soon. Thanks so much.
Thanks so much.
Take care.